Hi, everybody. I am super excited to be here. My name is Adair Borges, and I'm going to talk about some of my efforts at Berkeley and, at UC Berkeley and at Arcadia Science, searching for invisible phages in microbial communities. So phages are incredibly diverse and heterogeneous group of biological entities. And the phages that I'm most interested in are the ones that have really divergent features or super unexpected biology that makes them invisible to standard phage discovery pipelines and approaches. So first, I'm going to talk about my postdoc work in Jill Bamfield's lab at UC Berkeley, studying phages that use alternative genetic codes and are often invisible to standard pipelines that assume standard genetic code. Second, I'm going to talk about my work at Arcadia Science, which is a research and development company in Berkeley, California, looking at phages that use alternative nucleotides. And these are often invisible because standard sequencing assumes canonical nucleic acid chemistries. Okay, so when I joined Jill's lab in 2020, I was coming from like a purely experimental molecular genetics phage background. I had never done like metagenomics or bioinformatics before. And so in starting in this lab, I decided to do first a lot of reading to see like what aspects of phage biology would I be most excited to study with metagenomics. And when I started doing this reading, I found a set of papers which were super exciting to me because they all talked about this weird phenomena of phage using alternative genetic codes. And so prior to that, I had thought that there was just one genetic code and it had 3 stop codons, TAA, TGA, and TAG. However, in phages that use alternative genetic codes with recoded stop codons, one of these stop codons, like in this instance, the TAG stop codon is recoded to an amino acid like glutamine. And so when you have this situation where you have a recoded phage genome and you try and predict its genes in standard code, you get the situation where all the genes get fragmented by these internal stop codons, which I'm showing as stop signs. However, if you predict the genes in the right genetic code, now you can recover all these nice full-length phage orths. And so when I started in Jill's lab, I was really fascinated by this phenomena. I thought it was super cool biologically, and I thought it was also super cool informatically to figure out like how can we design a discovery pipeline to find phages that use alternative genetic codes with recoded stop codons. So Jill and I began by looking for phage-like contigs in gut metagenomes, so both human and animal gut metagenomes, and specifically looking for things that had really low coding density in standard codes. So I'm showing you one of these genomes here. And then I asked, can I fix these genomes by repredicting their genes in an alternative genetic code? So here I've repredicted the genes of this phage contig, except the TAG stop codon is recoded, and now we get these full-length genes. And so when we scaled this up and did this for hundreds and hundreds and hundreds of gut metagenomes, it was really cool because we were able to find hundreds and hundreds and hundreds of phage that use recoded stop codons. And so these phage are really diverse. They range from really small, so the smallest one is around 14 kb, to really big. The biggest one is above 600 kb. And most of these phages recode the TAG stop codon. However, some of them, which I'm showing here in green instead of yellow, recode the TGA stop codon. And what was really cool to me, like probably my favorite part of this study, was while most of the phage genomes we got appear to come from free phages, so phage particles, like I'm showing here, some of the recoded phages that I found were literally hiding in standard code bacterial genomes as prophages. So I'm showing you one of these hidden prophages here. In this contig, this is coming from a Prevotella genome, and I've predicted all the genes with standard bacterial, Prevotella code. And on the flanks of this contig, we can see like normal size looking genes. These correspond to the Prevotella host. However, in this middle of this contig, we see this like weird gene poor desert that corresponds to a sharp drop in GC content shown in this graph. So this doesn't look like much, but if I go in and repredict the genes, except using an alternative code where TAG is now recoded to be an amino acid, now we can recover all of these nice full-length signature phage genes and reveal this prophage that's like hidden in the bacterial genome. And so I really love this example for 2 reasons. One, it shows how standard code gene prediction can literally make alternatively coded phages invisible. Secondly, it clearly proves that recoded phages are infecting standard code bacteria that use a different translation system than them. And so as a phage biologist, this was super curious to me. I was very interested in figuring out why phages would use an alternative genetic code that should be incompatible with bacterial translation. And so canonically, phages use their host ribosomes and their host translation machinery to make their genes. And so if a phage is using an alternative genetic code with recoded stop codons, this is basically like the phage is filling its own genes up with stop signs. And so this should block translation. This should lead to, gene fragmentation, and this should ultimately be lethal for the phage. And so I took our large collection of alternatively coded phage genes, alternatively coded phage genomes, and looked to see if we could figure out what's going on here. And so when we looked at these phage genomes, we saw a pattern that had also been noticed by other people who had looked at these phage genomes previously, but that was that phages didn't actually use alternatively coded, stop codons randomly throughout their genome, kind of like I showed you on the previous slide. Instead, these alternatively coded genes were all clustered in one end of the genome, whereas basically the other half of the genome looked pretty normal. And so what we think is happening here is that when phages infect a cell, the early genes can be easily made with host ribosomes. However, all these structural and lysis genes, which are toxic, which are really bad if the phage expresses early, are filled up with these molecular stop signs. And so this is like a form of translational regulation where these genes cannot be produced until a phage is ready. And so when the phage is ready to produce its structural genes and produce its lytic genes, then it will produce a series of what we call code change molecules. So we see different ones in different phages, but basically we see suppressor tRNAs, aminoacyl tRNA synthetases, and release factors, which we believe should be able to change the code of the cell and now allow for these previously suppressed genes to be expressed, and the phage can produce its structural proteins, and the phage can also produce its lysis proteins, which will bust the cell open and release these phages to go start the next wave of infection over. So when I had started this project, I had assumed that it would always be bad for a phage to have its genes be incompatible with bacterial translation. But after doing this project, I realized that there actually are circumstances where a phage would want to prevent expression of its genes. And so we think that phages use this as a really clever form of translational gene regulation to prevent premature expression of lytic gene products. And so I learned a ton in this project. Like I said, I had never really used bioinformatics or metagenomics before to study phages, and this really profoundly changed the way that I thought about them. The two big things I learned are here. One is this very much emphasized to me something that's probably obvious to many of you here, but phages do things really differently. Phages are not bacteria, and tools built with bacterial rules in mind, or even built with the rules for model phages in mind, will break when they encounter phage, especially if that phage is like super weird. Secondly, we really need more cultured phages. To my knowledge, no one has cultured a phage with alternative genetic code. And that was one of the things that I wanted to do most during my postdoc, because, well, we can make models based on genomes and genes, predicting how this phage uses stop codon recoding throughout its life cycle. I really think that the next step in this project and the next step of understanding how phages use stop codon recoding will come from getting these phages in culture and studying them experimentally. So it was with these two ideas: one, that we need bespoke tools to study weird biology, and also that we need to get weird biology into the lab more, that I joined Arcadia Science, which is a new research and development company in Berkeley, California, specifically focused on the study of non-model organisms. So Arcadia Science was founded by Prachya Vasti and Sime Chow, two scientists, as well as Jed McCaleb, who is an investor and philanthropist. Like I said, the focus at Arcadia is our idea is that if we study non-model organisms, so the things that are like the exceptions to the rule or the things that do really weird stuff, we can learn a lot about the vastness of biology and the vastness of evolution. And in order to study non-model organisms, we need really good tools. So we invest really heavily in the development of technologies that are not focused on like, or not developed for like kind of like average model systems, but are specifically tailored to the non-model system at hand. Next, we are doing a lot of experiments in open science. And so we'll be publishing and sharing our work quickly and openly, but we will not be doing so in journals. We have our own publication platform on our website where we release our pubs and closer to real time as like more modular micro publications. And then lastly, we have money to do, discovery, technology, and community for the next 10 years. However, what we really want is to make Arcadia financially self-sustaining, so we can do it for like the next 20 years or 30 years. And so in order to do that, we are translating a subset of like our basic biology discoveries that we make studying non-model things into things that can be licensed or patented or even spun out to be new companies that can then fuel this next cycle of basic biology research at Arcadia. So when thinking about Arcadia science and the opportunity here, thought really carefully about like what aspect of phage biology, because basically all of phage biology is non-model biology, like what aspect of phage biology would I'd be most excited to like apply the Arcadia framework and resources to study. And I decided that I was going to study phages that use alternative nucleotides or have genome modification or use non-canonical nucleic acids. And so to do this, I've teamed up with Januka Athikorlich, who is an amazing phage biochemist. And so, as we've heard, phages have put extraordinary evolutionary pressure on bacteria. And one of the ways that bacteria fight back is by producing nucleolytic immune systems to chop up phage DNA. And it's been appreciated in the phage biology world for a long time that like one of the ways that phages can protect themselves from this is by using genome modification to shield their DNA from these nucleases or to stabilize it against the nucleolytic attack. And so this has been worked out in a couple of systems or in a couple of cultured phages, and it's super cool, amazing biology. But more recently, we've also come to realize that there are phage targeted immune systems that also go after RNA. So Januka and I are really excited about the possibility that maybe there are phage RNA modifications or phage alternative RNA chemistries out there too. So altogether, we consider it likely that like a lot of phages probably use alternative bases because bacteria have put so much evolutionary pressure on them to hide and guard their nucleic acids away with these nucleic acid targeting immune systems. So we think that there's this vast universe of alternative nucleic acid chemistry out there, but right now most of this universe is still invisible to us. So up until very recently, phage culture has been like the gold standard for studying phage genome modification. So get a phage in the lab, test its DNA chemistry and figure out exactly how this works. And these kind of studies have like laid the foundation for this entire field. However, we know most phages are not cultured. Like we're missing like vast sectors of phage biology when we just look at the things that have been brought into the lab. And furthermore, most phages that are cultured are not analyzed through this lens. So even for the phages that we know the most about, we don't know what their DNA or RNA chemistries are. So normally a question like, what is the diversity and distribution of a phage trait would be ideally suited for sequencing and metagenomics. However, sequencing chemistries like Illumina don't give you any information about the underlying chemistry of the genome you sequence. You just get a FASTA file back of A's, T's, G's, and C's. It does not tell you about the chemical state of the genome before you sequenced it. Secondly, to make matters worse, some of these really bulky, weird DNA can actually block amplification by the polymerases used in PCR amplification and Illumina library prep. So sometimes the coolest stuff in the metagenome actually is dropped out before sequencing. So PacBio works amazing for some modifications. And there's some really beautiful work coming out now from even some of the people in this room using PacBio to study phage genome modification. However, PacBio still relies on DNA replication, and so it's inherently not going to work for some classes of modification. And so last, nanopore, another one of our sequencing options, is theoretically great for this. It's a totally orthogonal way of sequencing nucleic acids, and it should be able to give you information about chemistry. However, nanopore only has models for a couple different types of modified nucleotides. And so nucleotides that are really different from those models will appear low quality to the base caller because it just does not have the ability to recognize and map them confidently. And so phages, which are going to be full of these modified bases, are just going to end up looking like really low quality reads and are going to be discarded. So my overarching goal is to help chip away at this problem and help measure the diversity and distribution of alternative nucleic acid chemistries across bacteriophages. And so in mapping out how I might approach this problem, I did a lot of reading, and I specifically got a lot of inspiration from papers of people who work with phages with modified genomes in the lab and have developed tricks and tips for figuring out how to crack open these phages and study their biology. And so I want to share one of my favorite papers here. It's from Michael Skurnik's lab in Finland, and it's about how they managed to sequence the genome of an unsequenceable Yersinia phage. So in this paper, this group got this Yersinia phage in culture. They were super interested in it. They wanted to study it, so they decided to sequence its genome. However, its genome was unsequenceable because it's modified, and so they were unable to get the genomic sequence for it. Then they did something really, really clever. Instead of just giving up, they decided to infect cells with this phage, harvest RNA over time, and then sequence the transcriptome. And so then they were able to reassemble the phage genome from the transcriptome, which is really smart. And so I love this paper because it teaches two important lessons. One is culture is really important. If they hadn't gotten this phage in the lab in the first place, it would have been very difficult for them to, throw the book at it and figure out some strategies for capturing its genomic information. Secondly, omics can complement each other. Even though one sequencing technique can fail for your particular application of purpose, when combined, they can create a more holistic picture and give you more information about the genomes in your sample. And so at Arcadia, I'm part of a team of microbiologists, Rachel Dunnan, Emily Weiss, and Manon Marin, who are very motivated and excited to figure out how to connect culture and omics-based techniques to create a more inclusive study of microbial communities. And our microbial community of choice is cheese. It's a semi-complex and delicious microbial community that is composed of bacteria, fungi, and viruses. And so it's kind of like a perfect substrate for developing different techniques, both culture-based and omics-based, to inclusively profile all the different kinds of biology happening in the sample. So on the culture side, my job is to culture all these cheese phages and then screen all their phage DNA and RNA for cool nucleotides. On the community level, we are working together to try and sequence everything. So sequencing the community, sequencing the virome, doing transcriptomics, doing metagenomics, and using both nanopore and Illumina, these complementary modes of sequencing to try and gain as holistic picture of these communities as we can via sequencing. So with Emily, we've been working together to pilot out some high throughput bacterial culture methods. And so we can get things like this, which is a 96-fold plate of arrayed bacterial isolates from our cheese community. And then I'll take viromes that have been prepared from that same cheese community and use both liquid and plate-based isolations to isolate cheese phages. And so these are some of my most beautiful cheese phages here. I'm super excited about them. And so after isolate all these cheese phages, you can sequence their genomes and chemically analyze their phage nucleic acids. And so on the community level, trying to figure out how do we use different types of complementary omics to find weird and missing phage genomes. I've been working with Taylor Ryder, who is an amazing bioinformatician, and we are trying to scale this approach of using RNA to find unsequenceable DNA phages. So we're searching for DNA phages hiding in metatranscriptomes. And so right now we're starting with doing a survey of a bunch of public data. So looking specifically for paired metagenome, metatranscriptome from a variety of biomes. And so what Taylor does is she takes the metatranscriptome and then she subtracts out the metagenome. And this is a graph of how much of the fraction of the metatranscriptome is left over after you subtract the metagenome. And it's probably really, really hard for you to read, but basically we see ranging from 44% down to 5% of the metatranscriptome is not contained in the metagenome. And so the next step is to take these leftover reads and then try and subtract out like all of GenBank and keep iteratively removing sequences until we get this pure transcriptome only fraction. And so for us, RNA phages are going to be our positive control. If we get a lot of RNA phages, we did our job right, because RNA phages definitely need to be in the metatranscriptome and not in metagenome. But what we're also hoping is we're going to see some DNA phage signature genes, and that those DNA phage genes will correspond to phages that have unsequenceable modified DNA, but can produce sequenceable RNA. And ultimately, we're going to be applying this regularly to our cheese samples or whatever communities we're bringing into Arcadia to do this as like a routine screen for unsequenceable DNA. And so in thinking about how to, make invisible phage visible over the past couple of years, here are my main personal takeaways. I said this before, but phages do things differently. They're not bacteria, and it's important to use tools that can deal with unexpected phage biology, like alternative genetic codes. Secondly, there's a ton of value in paired omics data sets. Even though individual types of omics can be faulty and biased against like one class of thing that you're interested in, when you put it together, you can often generate a more holistic picture of what's going on. And you can even exploit methodological biases to find cool biological signatures, like looking for DNA phages that are unique to the metatranscriptome. Again, I think we really, really need more cultured phages. Previously, I thought that metagenomics and culture were kind of like two ends of a spectrum, but I actually think that they really need to work together and have one inform the other so they can go back and forth to connect ecology and mechanism to find new biology, which is something very central to Arcadia's mission. And last, I think open science is the best science. So I've been profoundly influenced by, reading the papers and using methodologies and techniques developed by the phage community, developed by like many people in this room. And so it's a huge part of Arcadia's mission that we share our resources, that we share our data, that we share our findings quickly and openly and without barriers. So I want to end just by telling you about how you can continue to follow Arcadia's experiment and also my work on phage nucleic acids. So we have our own publishing platform where we very regularly post like small micro publications and protocols and data sets. This is the URL, but you can see like here's my project on exploring bacteriophage nucleic acid chemistries, and then here are other projects that are ongoing at Arcadia. And so the goal here is that people can like read and access our protocols quickly. They'll each have a DOI, so they can just be cited and used like a normal paper, but they should come out at a rate much faster. And there also is, has ability to comment and give feedback on something. So like if you wanted to use my sequencing data, but I didn't provide like usable metadata, you could just tell me and I would update my pub, and then you could proceed forward with something that's now usable for you. So that's our entire goal, is to make science maximally reusable, maximally transparent, and maximally advance the study of non-model organisms. So with that, I have a ton of people to thank. First, I want to thank the organizers for inviting me. I've had a great time these past couple of days. I want to thank the Banfield Lab for being an awesome place to learn metagenomics and learn all about like the weird parts of phage biology. I also want to thank my teammates and collaborators at Arcadia Science for being an amazing group of people to work with and to study phages with. And with that, I'll take your questions.
Fantastic talk. We have a couple of questions. time for a couple of questions. Okay.
Man, this stuff is the coolest stuff ever. I think this is really neat. What I'm trying to get my head around, though, is the antigenicity, if that's the right word, for these alternative DNA sequences, both from the perspective of the bacterium, but then also potential applications for this technology in eukaryotic hosts. Because I would think that, there's some papers that have looked at the, I don't know if parsimony is the right word, but the fact that phages tend to, use the same density of GC bases, for example, as the bacteria that make them. And I would think that there's a lot of pressures, both from an efficiency point of view and then again, antigenicity, if that's the right term, point of view to conform. Why aren't these conforming?
Yeah, so I think that phages kind of have like, or at least the way I think about it is phages have two options. One is to become as like similar to the host as possible, like, and be able to co-opt all of its machinery, like have a genetic code that's super similar to the host and therefore is compatible with ribosome, like have DNA polymerase that can be easily replicated by host biology. But then there's kind of like another route where there's selection for phages to develop things that are orthogonal to host biology. And so by having DNA that's super different from bacterial DNA, they're able to hide it away from bacterial immune systems and like factors in the bacterial cell that have evolved to recognize and bind and interact with DNA. And so I think that like, phages that are large, phages that can have a lot of genes like DNA polymerases and their own like nucleotide biosynthesis genes, like those are maybe more on like a route toward like autonomy. And then like smaller phages there might be selection for them to be more host compatible so they can rely on host machinery more to replicate them and produce their proteins, etc. So I don't know if that answers your question, but that's how I think about it.
We had one question from online. How does commercial private funding work for basic science?
How
does commercial private funding work for basic science?
Yeah, so we are super lucky in that our funders are primarily interested in the meta science experiment first, like the meta science experiment being like, if you just fund basic biology upfront and say like, we're gonna fund basic biology and we're gonna trust that like over a long time frame, like a time frame of 10 years, it will produce commercially useful things. Because we see this happen in universities. People do basic biology, and at some rate, people find really awesome, really useful stuff that can be patented, that can be spun out to companies, etc. So I think this is trying to like, we have funders who like understand this and are trying to recreate this cycle outside of academia.
Last question.
Thanks, great talk. I really enjoyed the latest section of your talk, which was all about studying the biology of some of the food products. So I'm wondering if Arcadia is also interested in like looking at things like kombucha, where you've got complex communities of yeast and bacteria that change over time due to acidification, et cetera, or maybe sauerkraut, kimchi, et cetera, and the health benefits of these kind of things. So what are the dynamics? But I can also see huge advantages of studying that from a psychological perspective of cooperation and conflict.
Mm-hmm.
And so I wonder if those are kind of within the mandate of the company you folks are working with.
Yeah, I mean, we chose cheese because Rachel Dunn has a ton of experience with it, and it's an incredibly tractable system in her hands and in the hands of people who have worked in her lab. And so we've started there, but like we definitely do not want to be limited to just cheese. So we're using cheese as kind of like our test bed to develop all these tools, but then would love to like port them over and try them in like another microbial ecosystem and see like, okay, how many tools break? Like, can we make the tools better? What new things can we discover? So yeah, like other fermented foods kind of fall in the same realm as like, they're gonna have their own complex biologies, but we can get them easily, we can get high biomass, we can start to test technologies on them. So I think like stuff like kombucha is like a great idea. And I would love to hear more from you if you have ideas about other cool communities that we could have on our horizon to study, because there are so many of them. And yeah, we're really excited to try our platform on them.